All right, thank you, Kurt. Like he said, I work at the USDA, and one of the main functions of our laboratory is to utilize agricultural products. So these are probably, I'm guessing that these are two crops that you have not heard of, and I wanted to compare biochars from the press cakes of these two products. I had two goals when I was working on this project. The first goal was to test pennycress press cake as a potential biochar feedstock for horticultural applications. More specifically, what we're looking at is to use it as like a peat moss substitute, peat moss vermiculite substitute in common nursery type plants that would be sold in greenhouses. The other goal I had with this project was to study process differences between 4 different pyrolysis methods. I'm very interested in different gasification and retort methods of making biochar, and what are the differences that come from changing certain parameters. So here's a quick review. I'm going to go through this in more detail later. The first, we have 3 gasification methods and 1 retort method that I used. The first gasification method is a high throughput continuous feed industrial gasifier, and this is the gasifier at Chip Energy, which is in Goodfield, Illinois. I know Paul Weaver. If you come to our meetings, the Illinois Biochar Group, you're probably familiar with Paul Weaver. He comes all the time. He has been great with supporting research in this area. This is a high throughput system. So you see this hopper is where you can put your feedstock. It gets fed with an auger into the bottom of the gasifier and then gets processed for heat. And then he has the biochar as a byproduct, and we take that and look at it. On a smaller scale, I also have some batch TLUD stoves. Paul Weaver also made these and sold these to me. I use these. You can see they're smaller. This is about this big, about 3 feet high and about maybe 9 inches across. These are nice. They work on the same basic concept as the one you just saw. They're just smaller and they're a batch. So you have to make your biochar, dump it out, and make another batch. This is the newest iteration that we have. This is the mini TLUD. And my colleague Steve Vaughn, who also works at the Ag Lab in Peoria, he designed and hand-built this himself. And we like this. I like this one in particular because it's smaller yet, but even though it's smaller, it's very convenient. We can run this in the hood in the lab. So we can keep cranking out lab-scale quantities, and it's very easy to work with. And last but not least, we have just your standard retort oven. It's a sealed nitrogen retort, and the advantage here is you can control the temperature very explicitly. So we can study temperature ranges and see what kind of effect that has. Okay, so why am I studying pennycress and Laesgerella? What are these materials? These are both oilseed crops, and we're going to start off talking with pennycress. Pennycress is a winter annual that can grow in existing soybean fields in the northern United States. So that's one of the big advantages is you can rotate this with soybeans. So you plant it after you harvest your soybeans. It overwinters on the field, reduces erosion of the topsoil, and this is good. They are working on trying to shortening the growing season for pennycress because it's not quite short enough to do this with corn. You can only rotate it with soybeans. But nonetheless, that's an advantage for soybean growers. And you can use this and have a winter crop that you can harvest. Why do they harvest it at all? Well, like I said, it's an oilseed crop. They're interested in the oil. There's a lot of good biodiesel potential from pennycress oil. My colleagues, we have a unit at the Ag Lab that works on bio-oils. They have looked at pennycress, and the ASTM methyl ester tests, which are basic tests for fuels, came out very positive for pennycress. It has very impressive cold flow properties. So its cloud point is negative 10 degrees Celsius. The plugging point is negative 17. That's really good. It's better than most soy-based biodiesel. So that's why there's interest in this material. Laesgerella this grows well in arid and semi-arid landscapes native to the southwest United States. So this, like pennycress, you see where these are nice crops because they don't compete with our major cash crops. Pennycress because it's a winter annual, and Laesgerella because it can grow in arid areas that you can't grow other crops in. The seeds contain hydroxy fatty acids that are very similar to castor oil, but they don't have the castor stigma. You may or may not know that castor plants are what you can make ricin from. So as you can imagine, the U.S. government is not real thrilled about people growing castor in the United States. So this is a substitute for that, which you can harvest castor oil. Also, Laesgerella has estolides, which enhance the flow properties of oils. So there's some applications here with like lubricant industries, cosmetics, things of that nature. Okay, and then finally, press cake. What is that? Well, press cake is basically exactly what it sounds like. It's the leftover seed meal after the oil has been pressed out. So this is a picture of a French press that we have at the Ag Lab, and your seeds go in here in this hopper. They come down, they're compressed, the oil comes out this little spigot right here, and then the press cake, once the oil has been mostly removed, runs down an auger this direction and gets dumped out at this end. This does still contain some residual oil. We're not extracting the oil from it. There's still some oil in there. So that's one of the things that we noticed is this stuff burns really hot in our gasifiers. When I ran pennycress, typically in our gasifiers, they will react at about 500, 600, 700 degrees. Pennycress was burning at about 1,000 degrees. It was really hot. I was curious if that was going to affect its surface area or getting all of those volatile gases off, if that would increase the surface area. We'll talk about that in a minute. Another interesting thing about press cakes is they're handy for our purposes because look at the shape here. This is what the press cake looks like. Since it's being augered out, it sticks to the auger, and you get these chunks that are curved, and they're about maybe a 1/4 inch thick. And that's nice because when you put this stuff in a gasifier, airflow is important when you have a reaction in a gasifier. You have to have airflow. So this stuff, because it's curved, it doesn't pack super tightly. So it has space for the air to get through, and that's very helpful for us. It's a nice material to work with. You can just dump it in and it reacts very well. Okay, I want to talk about some of the specific processing differences. One of the things I talked about is I want to study these differences in our different processing methods for making biochar. What are they? Okay, so for a retort system, of course you have a contained system. It's atmospherically controlled. You just have nitrogen in there. You've expelled all the oxygen. You have pure pyrolysis going on. That's great. Another nice feature is you can control the temperature. We can set the temperature for whatever we want. We can do different biochars at different temperatures and study their properties. The only problem with retorts that we run into is you do have liquids and gas. You have syngas and you have tars and things like that. Those are going to be stuck in that sealed oven. So that's kind of a mess to deal with. This is where I love having technicians because AJ gets to figure out how we get all the tar and stuff out of here using vacuums and cold fingers and stuff like that. But they can be problematic. Here's a little diagram of our temperature profile. When we do stuff in the retort oven, we run at 5 degrees. It's pretty slow. This is definitely slow pyrolysis. We run at 5 degrees Celsius per minute. We hold it at 1 hour. We did 6 different temperatures, 400 through 800 and also 1,000. And then it cools at 1 degree per minute until it's room temperature. So that's our process. This is all under nitrogen in a true retort system. Now I'm going to talk about our gasification systems. So the first one that I mentioned was the Chip Energy Continuous TLUD. When you see TLUD in the, I'll steal some of Paul Anderson's thunder. He is the expert at TLUD design. That stands for top lit updraft. And what that means is TLUDs are nice because you do not need nitrogen. You can run these in the atmosphere, run them in air. And the reason is because with the top lit updraft design, the combustion is localized to the top of the stove. So all the combustion happens at the top, and the feedstock is treated thermally on its way down. And so you get pyrolysis. That part of the feedstock doesn't have oxygen, but the top is where that oxygen stays. With Paul's system, it's nice because you have a continuous auger-fed system. You can process a lot of biochar. He can also control various processing parameters. I can't go into the details. This is proprietary. It's his device, but he can have some semblance of control, some parameters that he can change to look and alter his resulting biochar. This is designed as a heat source, and biochar is a byproduct. He uses this to heat his factory. And so it's handy because you do have that output. You can harness the energy through heat and also have biochar as a side product. Okay, let's talk about the batch TLUD. This is smaller scale. Again, it shares the advantages of the gasifier, so you don't need nitrogen. It runs in air. This is a batch system, a little bit more unwieldy to work with. We don't have a continuous processing of biochar. We have to do it batch by batch. No temperature control. We put our feedstock in there and we light it up, and it's going to burn at whatever temperature it burns at. This is a function of the feedstock. We can monitor the temperature. That's what these, I don't know if you can see these 4 little thermocouples that I've got stuck in there. That's how we measure the temperature and trace it to see how fast our reaction is going. We know when it's done. And this is also designed as a heat source with biochar as a byproduct. I bought this from Paul Weaver. These are cook stoves, so they are designed, they come with a thing you can put on the top, and you can put a pot on there and you can cook with it. It's designed for third world countries. It's a nice cheap way to have a heat source and cook things. For us, our purposes, this is a little bit inconvenient because I have to get this room reserved. It's nice that we can run it indoors. We have a place, it's our solvent disposal room. So it has all these giant vacuum trunks that really draw a lot of air. So that's handy. I can run them under the vacuum trunks and run them inside, and it's okay. But I have to set it up and make sure that our solvents are taken care of. It's a little bit inconvenient. I have to make plans to use this thing, and that's one of the drawbacks. The mini TLUD is great. This shares all the same commonalities with the batch. No nitrogen, it's a batch system, and no temperature control. But the nice thing about this is it's designed to produce biochar, lab scale amounts of biochar, and it's more convenient. This is small enough where we can run this in the hood or outside. So that's a big plus. I don't have to reserve the room. I don't have to go through all that rigmarole. We can just do this on a quicker scale. Okay, so let's talk about some results. Again, I apologize too. This has got a lot of small print on it. What we've got here is an elemental analysis of the biochars that we made. So over here we've got the feedstock. At the very top, this is not biochar at all. This is just the raw pennycress and the raw Lescarella press cake. And then we've got ash in this column: carbon, hydrogen, nitrogen, and oxygen. Here are our different procedures. We have our retorts at our various peak temperatures, the batch TLUD, the mini TLUD, and the chip energy TLUD, and then the same thing for Lescarella. So let me draw your attention to a few different things here. As I mentioned, the top is the raw feedstock. You notice that Lescarella has slightly less ash. Otherwise, they're pretty similar. Slight difference in oxygen and hydrogen, but basically carbon is very similar. So very similar starting materials in terms of these properties. One interesting trend that we noticed is that you notice with the retort series, as we go up in temperature, ash increases with temperature as well. You see a definite increase all the way up to 1,000 degrees. That's one interesting trend we see. Temperature is going to have a play here too. I didn't want to make this graph any more cluttered than it already was, but temperature will have an effect on the batch, on the other TLUDs as well. Let's talk about carbon. Carbon, we did not see an overwhelming trend. If you look, these values all kind of fluctuate around 58% plus or minus 4% or so. So we did not see any distinct trends with carbon. If you look at both pennycress versus Lescarella, you could say that it appears that Lescarella has slightly higher carbon content than pennycress. And carbon content is one of the things that we're worried about, or I shouldn't say worried about, but one of the things that we pay attention to, because this has an application in our potting mix products. Another thing we're very interested in is surface area. So surface area affects your water retention capacity and things like that. So one of the things that I want to point out is in this one, we've got surface area is on the x-axis. So higher surface area, you see a bigger bar to the right, and then we've got all of our different processing methods are on the left. Here's the retort ones at the top at different temperatures, the chip energy TLUD, our batch TLUD, and the mini TLUD. The only other thing on this graph is green is pennycress, orange is Lescarella, and the little dotted section here, that's micropores. We can also measure the micropore concentration of the surface area, the micropore component, and so that's also reflected on this graph. So one of the things I want to talk about here is you can see the importance of temperature. Here in our retort series, you see how we have very poor surface area at 400 degrees centigrade. As we go up, draw your attention to the green bars. If you look at pennycress in green, you see that there's kind of a peak at 600, and then it starts to drop a little bit. If you look in the literature, there's some pretty good hypotheses of why this happens. They feel that as you increase the temperature, you're burning more and more volatiles out and cleaning out all that space, you're making it more porous. But when you get to a high enough temperature, you start to actually break down the biochar. It breaks down and it starts to crumble in on itself, and some of those pores get sealed up again, and that's why you see that drop. Another comment I want to make is just overall, with pennycress and Lescarella, our highest surface area was 140 square meters per gram. That's okay. It's not great. Activated carbon has, between like 1,500 to 2,000 square meters per gram. It's got incredible surface area. We've run, in past research, we've looked at a lot of hardwood biochars that have 400, 500 square meters of surface area. So it's okay, but it's not great. And I think the difference there is this is a seed product. This is a crushed seed. You don't have the wood with the xylem, and you don't have that structure inherent in the system with these because they're seeds, they're crushed seeds. We do notice that in terms of surface area, the mini T-Lud does really well for Lescarella, not so much for it does really well for pennycress, not so well for Lescarella. Lescarella seems to work better in the T-Lud, which I will say is significantly higher temperature than the mini T-Lud. Okay, so let's look at some other chemical properties. This is a really important part of the study when we're talking about our peat moss and the response in growing things. As was mentioned before, most biochars are very high pH. We see that as well. Almost all of our biochars that we produce have a pH of 8 or 9 in that neck of the woods. They also have a very high electrical conductivity due to potassium, and this leads to poor horticultural potting media. Let me take a minute now and explain what the experiment is to look at this in potting media. This is the experiment that Steve Vaughan designed, and we know one of the things I want to say is one of the greatest misconceptions I think the public has about biochar is that it's fertilizer. Biochar is not fertilizer. It needs an organic component to really make it effective if you're going to grow stuff with it. So what Steve designed was he thought it would be great to couple organic potato digestate as the organic component with an inorganic component, in this case biochar, as a substitute for the peat moss vermiculite potting mix. So our control was peat moss and vermiculite as organic and inorganic, and then we used potato digestate as the organic component, and then we looked at 3 different biochars as the inorganic component. We looked at wood pellet biochar, wheat straw biochar, and the pennycress biochar. And what did we see? We saw a lot of stuff, and we got a lot of data. This is just a few select things that I wanted to point out that I think are the key points. Hopefully you'll be able to read about this in a few months. We did submit this to Industrial Crops and Products, so hopefully that will get published in the future. The one thing I wanted to point out was here, the pH here, they're all the same at 6. And that's because these are pH adjusted. One other thing we added to the system was Steve used citric acid to adjust these down to pH 6 so we can compare them all on an equal pH basis. What I've got here is I've decided to show you pH, electrical conductivity, and the potassium concentration of the wood pellet biochar, wheat straw biochar, pennycress biochar, and then this is representative of a typical potting mix. So this would be close to the control. This would be typical values you would see for the control. So pH is 6. That falls in line. That adjusts it basically to what we would hope to get with peat moss. The electrical conductivity for wood pellets, 1.7, wheat straw, 3.7, and pennycress, 8.5. It's really high. It's too high for a typical potting mix. The reason that is so high is because of potassium. As you notice, for pennycress, potassium is just through the roof, 5,000 parts per million. And in fact, when you look at the typical potting mix, even wheat straw and wood pellet are pretty high with respect, but we'll see if that affects. So the question is, so that's the results in numbers. How does this affect plant growth? When we did plant studies, Steve measured several different things. We did plant height, and then he would take the plants out after a certain time period, weigh them when they were wet, and then get as much, do a dry weight as well. Here's the data. So we looked at 2 different plants, tomato and marigold. Over here we've got our sample. Here's our control sample, peat and vermiculite, the potato digestate and wood pellet biochar, wheat straw biochar, pennycress biochar. We've got plant height, the fresh weight, and the dry weight. So let's look at tomato first. Here's the control, and you notice that the wood pellets and the wheat straw did fairly well. They're higher, the weights are higher, and the weights in all cases are higher, and the wood pellet biochar did really well. It's significantly higher than the control. The pennycress, not so much. It's about the same height. You lose a lot of weight, and I think that's due to the high potassium and the high electrical conductivity. For marigold, we saw similar results, except in this case, it shows, this is just an illustration of what we've been talking about, that every biochar is different, and every system is different, unfortunately. It's not like we can apply these to everything. But for the Marigold, you note that in this case, the wheat straw biochar did pretty well in terms of plant height and dry weight. And again, we see for the pennycress, not so good. So unfortunately, the bad news is pennycress is not a good candidate for this, but the good news is we did find, we ran 2 other candidates that seemed to work very well. Now I had time, one other interesting result that we had was with the surface area. My colleague Mike Jackson at the Ag Lab does surface area studies and runs these numbers for me, and we use the BET method, which is a pretty well-known method to determine surface area. And what happens in a BET experiment is you have your biochar in a chamber, it's vacuumed, you get everything out of there, and then you add a very small amount of, usually nitrogen, you can use carbon dioxide, a small amount of gas, and then you measure, you have a very sensitive pressure transducer. And generally, what you see in these types of plots is you see a straight line. As you add gas, you see a straight line and the volume goes up, and you can trace this. And what he saw for a couple of my Lescarella biochars was this. And this is an interesting shape where it goes up and hits a point, and then the slope changes drastically. What that means, and don't ask me any questions about this because I don't know the details of the BET method. I'm not an expert in BET method, but Mike says that what this means is you have a very monodisperse pore size in this material. So this inset plot is a plot of the pore size in nanometers, and you see a very narrow spike at 2 nanometers. So we've got a biochar that we did in a retort furnace that has very uniform pore size. This is very interesting, and here's another one. Here you get a less drastic slope change, but you see how the slope changes right there. We add gas and it goes in and the pressure doesn't change, and then boom, you hit a certain spot and the pressure stays constant because you're filling all those pores. And then once they're filled, then the pressure starts going up again. So we have a very uniform 2 nanometer pore distribution in this material. This caught my eye because I have a colleague at the Ag Lab, Mike Appel, does a lot of work with pulling toxins out of like apple juice or wine or other food products in the food industry. And he designs synthetic polymers with a certain pore size so that they can pull certain toxins out. If you have a certain pore size, they're very good at pulling toxins out. And I said, hey, Mike, I've got this material that naturally seems to have a very monodispersed pore size. Could you look at this? So this is something that we're looking at. We're in the process of studying, and that could be something very interesting. So let me bring you to my conclusions. Comparing the pyrolysis processing methods, in terms of carbon content, we had little change between the methods. There wasn't a drastic change. Lescarella was slightly higher than pennycress, and for pennycress, the CE T-Lud was the best, and the batch T-Lud was the worst. For ash content, we noticed that you definitely see a trend with temperature. Ash content directly increases with temperature. For surface area, we seem to see a peak around 600 degrees Celsius for the retort series, and the Chip Energy T-Lud was very poor in terms of surface area, and the Mini T-Lud did well for pennycress. Now for my other goal, let's talk about pennycress as used for a potting mix. The pH and electroconductivity levels were very high, unfortunately. We thought one possible activity, maybe you can use this for acidic soils. Since it's very high pH, you may be able to treat acidic soils. And the surface area is moderate. It's not great, it's not horrible, it's in between. Unfortunately, the potassium levels rendered pennycress biochar to perform poorly compared to biochars from other feedstocks for tomato and marigold growth, but we do have those others that we're more hopeful for. And so with that, I'd like to thank my coworkers, Paul Weaver at Chip Energy, for running those samples for me, Steve Vaughn for doing all of the horticultural studies, Mike Jackson does the BET stuff for me, AJ, Ashley, Judy, and Ray are the technicians at the Ag Lab that provided all this data. And last but definitely not least, I want to thank Nancy and Beth for doing so much work to organize this conference, and so we can all be together and learn something today. So thank you.